Hi, now I am going to talk about electrophoresis. Electrophoresis is the name derived from a Greek word which means to bear electrons. This technique was first introduced by A. Tiselius in 1937. The electrophoresis is the separation of charged molecules by applying electric field. Electrophoresis is applied to separate DNA, RNA, and the protein molecules. Under the influence of electric field, these charged molecules will migrate either to the cathode or to the anode. The nucleic acids such as DNA and RNA molecules have negative charges. They migrate towards the anodes. And the proteins are positively charged molecules. They migrate towards the cathode. The rate of migration of these charged molecules are depending on the net charge of the molecules, the shape and size of the molecules, the pH and the composition of the supporting medium, the strength of the current which is applied, and the temperature of the operation under which the electrophoresis is carried out. And the electrophoresis, some basic requirements are there. They are: first one is the electrophoresis unit, which includes 2 buffer tanks and 2 platinum electrodes, buffers and supporting medium and the samples. Next is the electric current, that is the power supply. The buffers in electrophoresis has 2 functions. First, the buffers keep the medium at constant pH. It also helps to carry the applied electric current. The buffers will determine the type of charge on the solute and the ionization of the solute and the electrodes towards which the solute will migrate. Some commonly used buffers are phosphate buffer, tris-borate EDTA, it is otherwise called TBA, and tris-acetate EDTA, that is TAE, barbitone buffer, and tris-glycine buffer. Next is the supporting medium. The supporting medium on which the samples are loaded. It is required to separate the molecules according to the size as well as their mass, etc. An ideal supporting medium should contain high transparency, low adsorptivity, desirable sealing effect, low toxicity, high electrical conductivity, controlled porosity. Chemically, they should be inert and easy availability. Based on, for example, the paper strip, cellulose acetate, agarose, starch, polyacrylamide, and the Cephadax are some of the supporting medium here. Based on the types of the supporting medium we are using, the electrophoresis are classified into different types, that is paper electrophoresis, agarose gel electrophoresis, polyacrylamide gel electrophoresis, sodium dodecyl sulfate polyacrylamide gel electrophoresis, that is SDS page, pulsed field gel electrophoresis, that is PFGE, and 2-dimensional electrophoresis 2D. And this figure shows the paper electrophoresis. Here the 2 buffer tanks are there, and the filter paper is placed in between these 2 buffer tanks, and 2 electrodes, that is positive electrodes and negative electrodes from the power supply. And this figure shows the agarose gel electrophoretic unit. Here two buffer tanks and the supporting medium, that is the agarose gel. It bears the well for the samples and the power supply. And this figure represents the polyacrylamide gel electrophoresis unit. Here the samples are loaded at the top. The larger molecules with high molecular weights remain at the top, and the molecules with this low molecular weight moves at the opposite end. And this figure shows the SDS page setup. And this is the diagrammatic representation of PFGE. Here the electric field, electric current in alternating ways, that is, it is from left to right and right to left and top to bottom, like that. It is altering every time. And this figure shows the 2D electrophoresis setup. First, the protein molecules are separated based on the isoelectric point. Then the separated molecules in the tubes are then transferred to the SDS page setup. Here the protein molecules are separated like spots. Then coming to the applications of electrophoresis, the electrophoresis are used to separate protein, DNA and RNA. It is also used to determine the molecular weight of the protein and the nucleic acids, and used to determine the size of the DNA fragments. It also used to resolve thousands of proteins and DNA in the biological samples. Then we can see the application separately for each and every types of electrophoresis. First one is the applications of paper electrophoresis. The paper electrophoresis is used to distinguish the solutions which are containing micromolecules in different proportions. It is also used to demonstrate any change in charge of the molecules in return to the pH change. It also used to separate the small molecules, that is biomolecules, in solutions. It is also used for the serum analysis for the diagnostic purpose, can also be carried out by the paper electrophoresis. Next one is the application of agarose gel electrophoresis. The agarose gel electrophoresis is used for the separation of proteins and DNA fragments in the samples. It is also used to detect the purity of the DNA fragments. The agarose gel electrophoresis is to determine the size of the DNA fragments. Next is for the application of page. The page is used for the separation and purification of the protein and DNA fragments in the biological samples. Also used to separate proteins based on the charge but not the mass of the proteins. It is used to determine the molecular weight of the DNA and proteins, and also used for the PCR analysis. The page is also used for the DNA sequencing process. Next is the applications of SDS page. The SDS page is used to separate proteins based on the mass as well as their size. It is used to determine the molecular weight of the protein, and also used to separate different subunits of complex proteins. The SDS page is used to resolve thousands of proteins in a biological sample. Next is the applications of PFGE. The PFG is used to resolve DNA molecules of 100 to 1000 kilobase pairs. It is used to distinguish the smaller fragments from the larger ones. Next is the applications of two-dimensional electrophoresis. The 2D electrophoresis is used for proteome analysis, cell differentiation, and detection of disease markers, monitoring therapies, drug discovery, cancer research, purity checks, and microscale protein purification. Thank you.